Okay. The goal of this screencast is to review what happens when your body mounts an immune response and be able to describe this in terms of how it leads to long-term adaptive immunity, the type of immunity that is specific to specific pathogens, and also create cells that remember those pathogens into the future, in a sense. And then be able to relate the different blood cells to their respective roles in this immune response. And this involves a pretty complicated figure that I've busted up into a bunch of little sub-figures so that you can bite it off into little bite-sized chunks. So interesting enough, the Nobel Prize this year in medicine was awarded to people who were looking at how a body can essentially protect itself from foreign invaders while also not destroying itself through autoimmune diseases. And this just happened to be an announcement that was made while we were going through this process of thinking about immunity. So I found that to be interesting. So when we think of the immune response, we can divide the immune response into what we call the humoral immune response, which is that having to deal with antibodies to organisms that are infecting you. And then we have the cellular immune response. And in the humoral immune response, a virus is phagocytized and deconstructed by antigen-presenting cells. And so in the case of SARS-CoV-2, which is the COVID-19 virus, this is a virus that has a capsid, and that capsid is encapsulating the RNA. And on the outside of this are a set of proteins called the spike proteins. That's what these blue things are. And so in the bone marrow, you have cells like B lymphocytes, and these B lymphocytes are involved in humoral immunity. And these B lymphocytes can come into contact with SARS-CoV-2, and when they come into contact with it, they come into contact with the spike protein, and they can then become sensitized to the spike protein. But another route by which this can happen is antigen-presenting cells, things like macrophages, can basically come into contact with the virus, dismantle the virus, and then translocate the spike protein onto the surface of it. And then it can present that virus to other cells. And the other cells have receptors on them called the histocompatibility proteins. And these histocompatibility proteins are basically the proteins that are involved in self-recognition, but they are also involved in keeping track of antigens that get presented. And so these macrophages will present these antigens to the T cells and the B cells, the T lymphocytes and the B lymphocytes. And so in the thymus, you have the production of helper T cells. And this macrophage is going to present these antigens that it has picked up from the virus to the T cell. And then these T cells will come over here and make contact with B lymphocytes, and it will basically activate the B lymphocytes. And when that happens, these B lymphocytes differentiate into what are called plasma cells. These plasma cells are the cells that produce the majority of the antibodies. And these antibodies then will go and latch on to the antigens of the virus, because these are now plasma cells that have been sensitized to these spike proteins, and so they produce antibodies that are specific to the spike protein. Those antibodies bind to the spike protein and essentially neutralize the virus. If the virus has all of its antigens, the spike protein, taken up and bound by these antibodies, it can never come into contact with ACE2, the cell receptor that basically invites the virus into your host cells. The other thing that antibodies can ultimately do is they can destroy and decompose the virus, and that once again ends the viral infection. So the first route of defense is the production of antibodies circulating in your bloodstream that neutralize the virus and kill the virus, so that the virus is destroyed, or the virus can't interact with the receptors on your cells that cause it to be invited into your cell in a vesicle that then prompts your cells to create more copies of the virus. The other thing that happens with these helper T cells is these helper T cells will represent antigens to B lymphocytes. So these helper T cells will present the antigen again to other B lymphocytes, and these B lymphocytes will then become memory cells. And so we have memory B cells and memory T cells, and these interact because they're essentially representing the antigen to B lymphocytes and to themselves. And this is where you get your long-term immunity. These memory cells essentially remember, in a sense, the fact that it has come into contact with things like the spike protein, so that if it ever comes into contact with them again, it can very quickly differentiate into plasma cells and mount an immune response without all of this presenting of the antigen stuff at the beginning. These memory T cells, though, can also differentiate into cytotoxic T cells that are also long-term and remember the fact that they have come into contact with the spike protein in the past. Cytotoxic T cells are also formed from the thymus. So cytotoxic T cells come from the thymus, but these are referred to as naive. All that means is that they haven't come into contact with a antigen at this point. Once they are produced, they will interact with macrophages. Once again, this is just this macrophage up here that is now down here. This macrophage will present to that cytotoxic T cell the antigen, which is this little red dot right here, that is bound to the major histocompatibility complex. And they will also interact with helper T cells that produce chemical compounds called cytokines that help activate the cytotoxic T cells. And these also interact with the macrophage. This whole process essentially activates the cytotoxic T cell to basically become active. This activated cytotoxic T cell, and I'm going to move my picture of myself, this activated cytotoxic T cell then interacts with infected cells, cells that have been infected by the virus, and it causes those cells to undergo apoptosis. Apoptosis is simply called programmed cell death. It's basically a set of chemical signals where the cytotoxic T cell tells the infected cell, you need to dismantle yourself in a programmed and systematic way. And this programmatic, systematic way of doing this is called programmed cell death or apoptosis. And so essentially, this cytotoxic T cell signals through chemical signaling and receptor proteins, signals this infected cell to dismantle itself. That then makes it no longer a factory for putting out new virus. One of the results of this is it also kills the cytotoxic T cell. And then finally, we have natural killer cells. These natural killer cells, once again, induce programmed cell death in virus-infected cells, but it doesn't require the interaction of these histocompatibility proteins for this to happen. So remember, we had antigen attached to histocompatibility proteins here that interacted with the cytotoxic T cells. In the case of natural killer cells, they're formed in the bone marrow. They have a receptor on them, and that receptor can actually interact with the spike protein directly on these infected cells. And once it does that, once again, it creates a chemical signal that induces apoptosis in the infected cell, and the natural killer itself also undergoes apoptosis as a result of this process. So this is the first infection. When you're first exposed to SARS-CoV-2, the COVID-19 virus, your immune cells basically embark upon all of these things happening. Your humoral response produces antibodies and memory cells that then can differentiate into antibody-producing cells. And your cellular immune response also kicks in, once again, activated by exposure to the antigen from the virus to basically hunt down infected cells, cells that have been infected by the virus, and tell them to kill themselves, essentially. But then what happens is if you come into contact with this virus again after you've been in contact with it once, you enter into a secondary response. And this secondary response essentially involves activating these memory cells, memory B cells and memory T cells. So now you come into contact with COVID again. Immediately, your memory B cells and memory cytotoxic T cells will differentiate. Memory B cells will differentiate very quickly into plasma cells, and immediately, boom, you're making antibodies and you're immobilizing and destroying virus. In the case of the cytotoxic T cells, you don't have to go through all these intermediate steps. These cytotoxic T cells are ready to make copies of themselves and start killing any of your cells that have been infected by the virus. And so the end result of this is that you get a much quicker antibody response and a much quicker cellular response. Similarly, the natural killer cells are also hanging around such that when they come into contact with cells that are that have the spike protein on them, once again, they can also induce apoptosis. And so anytime you have a secondary encounter with some sort of pathogen that you have previous experience with, the immune response tends to react much more quickly than your first response because it doesn't have to go through all of these beginning and intermediate stages. The immune system is already primed and ready to go. And this is why you get a vaccine. When you get a vaccine is basically exposing you to the antigen on the outside of the pathogen without the pathogen being able to make you sick, in theory. That then prompts this initial immune response, sets up memory cells that are then ready for any sort of secondary exposure that you get. In the case of COVID, you go and get your COVID shot. It starts this pathway down the first immune response so that if you come into contact with COVID again, it basically is a much faster response and helps you from either getting sick at all, or if you do get sick, it's much less severe than if you were seeing COVID for real as your first response. This is also why we get boosters occasionally. In some cases, you will get a vaccine, and then after a year or whatever, they will ask you back to get that vaccine boosted because the first dose of vaccine started this initial immune response, and the booster basically activates these memory cells and it builds up your immunity. It builds up this bank of memory cells. And so this is how vaccines work. They work by providing you with an immune stimulus that prompts this initial immune response so that if you ever do come into contact with the actual organism that causes you disease, you can mount an immune response much quicker. And I think that is the end of this. Yeah, so when you come into secondary response, it's just this part of the immune response that is being activated. And now I think that's the end of that. So that is that, and we have one more relatively brief screencast, and then this will basically kind of finish things off.